So to start, this is going to be the Exam 2 review session that covers skeletal, joints, muscle, and nervous. We'll start with the lecture component. So same thing as the last exam, there's going to be 50 questions. Of the 50, you're going to see 40 multiple choice first, and then the last 10 are fill in the blanks. And they are divided into the chapters. So there's 4, actually muscles technically is 2, and nervous technically is 3. So the points are going to be divided like 8 questions here for skeletal, 8 questions for joints, and then the rest are divided amongst muscle and nervous. Okay, so 50 questions, 40 multiple choice, 10 fill in the blanks. You will have 50 minutes unless you qualify for extended test taking time. And if you do, make sure that when you go and check on Canvas that it'll tell you how much time you have on it. So make sure that you have been given it. You should already, because I went through them. But in case something happened and it didn't work, let me know right away. Okay. So otherwise, everyone has 50 minutes to take the exam. You can take it anytime between starting midnight on Sunday. Think of it as like 11:59 Saturday. You have until Monday, 11 p.m. Central Standard Time. Now I know there's been some internet issues because of the weather, so just I really highly recommend not waiting until last minute to do it, because if you wait too last minute, it doesn't give us time to check in to see if there's something wrong with your internet or with the system. Okay, so just because of the increase in technical issues, try to take it before like 10 p.m. on Monday Central Standard Time. Okay. What else? Any questions about the lecture component? Okay. Then for the lab, it's going to be 30 questions. It's going to be divided between the bones, the muscles, and all of the models for the nervous system. That's going to include the eye, the ear, the brain, the spinal cord. So those 4 things are going to be on the lab. There are 30 questions. You have 45 minutes to do this one. And all of the questions are type in. Now, for the images, always look for the question is going to say what is this image, or what is this arrow pointing to? So if you see an arrow, always look for where the arrowhead is touching. That's what it's asking for. Another thing you can do for the lab is if the image is too small, if you press Control plus, it will zoom in, and you can press Control minus to zoom back out again. Okay. So that's for the lab. Spelling counts. Make sure all the terms are in there. For example, if you're identifying one of the carpal bones, you know that to do the carpal bones, you have to have the position, the name, the number, if there's a number, and whether it's a phalanx or not, phalanges or not. So just however the terminology appears in your lab PDF, make sure that you write it according to the same thing. Let's see, what else? Spelling counts, 45 minutes. Okay, questions on the lab? Okay. So I am working with your TA to make sure that she's able to go through your environmental scan to make sure that you are doing appropriate environmental scan. So she's going to be working on that for the first exam and for this exam. So we're still trying to catch up on that. But make sure that you understand that you have to use Respondus Lockdown Browser. And as a part of the Respondus Lockdown Browser, it's going to require you to have a webcam on. And then to start the exam, you have to do an environmental scan. Okay, and your environmental scan, I have tons of directions as to how you should do it. A lot of you are doing a magnificent job, but the few that I've already watched, there's a few, like one or two, that are not doing it correctly. So I'll be contacting you to make sure that you are doing it correctly. Because again, it's for your benefit more than anything else. Okay. Any questions on the logistics of the second exam? Okay. So I'm going to focus on the two content that was requested by your peers, and that's going to be the muscle contraction and muscle overall, and then neurophysiology. Okay, but just to make sure you know, when you're reviewing for the skeletal, make sure you know the types of bones. So, for example, what kind of bones are short, flat, irregular, and long. And of these 4 types, we clearly focus on the long bones. Okay, then and that's why later on when we go through anatomy, it's the anatomy of a long bone that you want to make sure you're able to know the structure and the function for. Make sure you know the functions of the bone, which include movement. Now, the bones by themselves can't move, so they have to work in conjunction with the muscles. They allow for stability by creating a framework for everything to hang on, like your skull is a framework, your ribs are a framework, and your pelvic bone, okay? That's why another function is protection. Bone contains bone marrow, and because of that, you'll find red blood cells being produced there. And another function of bone is that it can store minerals. And what minerals usually are stored in bones?
Calcium.
Yep, calcium phosphate. Yep. All right. So anatomy of a long bone, make sure you know the structure and the function. A really good way of doing this is just to draw it and label. So what would you call the ends of a long bone? The ends of a long bone. What's the ends of a long bone called?
I'm not sure if I'm saying this right, but the epiphysis?
Epiphysis, excellent. Yep, the epiphysis are the ends of a long bone, and then the shaft of a long bone is called what?
Diaphysis.
Mm-hmm. Diaphysis, yep. So that's the shaft of a long bone. Then in between the epiphysis and the diaphysis, you have this structure. What is that called?
The epithelial plate?
Yeah, epiphyseal plates. They are also called the metaphysis, or a layman's term, which you don't want to use on the exam because the exam always asks for technical terms, right? Is the growth plate. And that's how you can tell whether, if you were into forensic science and you found a set of bones that still had the epiphyseal plate, you know that they are what? An adult or a child?
Child.
Exactly. Yeah. So the epiphyseal plate gets stimulated at puberty, and it starts out as cartilage, and it converts to bone, and that's what allows people to grow taller, to elongate. Okay. And that gets stimulated at puberty. Okay. Wonderful. Then in the center, number 4.
Bone marrow.
Excellent. Oh, it contains bone marrow, but what is that space called?
Medullary cavity.
Perfect. Yep. And the reason why I'm asking for a response is because if you say it out loud, you're more likely to remember it. So if you struggle with the pronunciation of it, you're less likely to remember. You're like, what is that called? I can't remember, right? So even if you pronounce it wrong, as long as you're saying it out loud, it actually helps your brain to process the term and helps you to remember it. So even if you're saying it wrong, and there's really, you can say it wrong, but honestly, depending on where you're from, we say things differently. Like, for example, I had a professor that says respiratory, and another professor that calls it respiratory. So I don't care about the pronunciation, but the more you say it out loud, it helps your brain to process what it is, as well as understand it a little bit better. Okay, so don't worry about saying it wrong. Just say it, and I'll fix you if it needs fixing. So the medullary cavity, and the medullary cavity is, you're right, is where you'll find the bone marrow. Now, bone marrow is really interesting. It starts off as red bone marrow, and it actually transitions to yellow bone marrow as you become older, and then eventually it becomes gelatinous bone marrow. Okay, and we're in the elderly, but that's okay, right? Because red bone marrow is where the red blood cells are made, but that's okay because you still have in the epiphysis, you have the trabecula, and actually that's where your consistent red blood cells are being produced. Okay. So although the bone marrow transitions from red to yellow to gelatinous, you still have red blood cell production throughout your whole life because that's not the only area where you find bone marrow. Okay. All right. Now we're going to look at coverings at this point. Okay. So the coverings, the connective tissue that covers around the epiphysis, what is that called?
Compact.
I'll tell you what its function is. It prevents bone-to-bone friction.
Cartilage.
Yep. What is it called though? It is made up of cartilage. You're right.
Articular cartilage.
Perfect. So the articular cartilage that surrounds the epiphysis of long bones, when two bones articulate or when they move, they can potentially rub against each other. So the articular cartilage prevents bone-to-bone friction. Now, as you age, unfortunately, a process of aging is wear and tear. So the articular cartilage starts to wear down, and eventually the bone rubs against bone. And that's a degenerative condition seen in elderly people, and they start to have arthritis. So that's where arthritis comes from. Okay. So that's found at the ends of a long bone. Right. Then we have this connective tissue that surrounds the diaphysis. What number we got? 6. What is this called? And it's interesting because there's one on the outside layer, and then there's actually one on the inside. We'll start with 6A, the one on the outside, and then we'll go into 6B. What's 6A called? The one on the outside. What surrounds the diaphysis of a long bone? Give you a clue. What's the term for around? How about this? What's the term for bone? Okay. What's the condition? Yeah, go ahead.
Periosteum?
Yes. Peri meaning around, osteum meaning bone. And then the one on the inside would be endosteum. And you learned about the endosteum, right? Because that's where you will find what type of bone cell that allows for repair and healing. So let's say you fell and broke your bone. It's going to heal from the outside inward. It starts from the endosteum and then moves inward. And because what cells will you find in the endosteum? I'm going to list some cells. You tell me when to stop. Osteoblast, osteocyte, osteoclast, osteogenic. It's one of those four. That's all there is. Which one would you find the endosteum? Make a guess. Completely okay to get wrong. Which one?
Osteoblast.
No, but that's great because studies show that if you answer wrong, you're more likely to remember on the exam. Now answer it correctly. So keep going until you get the right answer.
Cyst.
Nope. Keep going.
Class. Osteoclast.
Keep going.
Whatever the first one sells. I can't remember what you said.
Call a classmate.
Osteogenic.
There you go. Yep, osteogenic. See, so now you're going to remember when you see it on an exam, right?
Hopefully.
Yes. Yep. Yeah, so you find osteogenic cells in the endosteum, and that's what helps to repair broken bones. Okay. Then, let's see. I feel like there's one more structure. I think that might be it. Epiphysis, diaphysis, epiphyseal plate. I think that's it. Yep, I'm happy with that. All right. So if I were to take a cut of this, cut it in half, and you're looking downward view of it, you're gonna see something like this. I'm gonna keep the color. There's this. And then here's this. So remind me what this is again.
Osteum.
Nope, I just took a cut of the bone. So that was 6A. What's 6A again?
Periosteum.
Mm-hmm. This is 6B. What's 6B again?
Endosteum.
Yep. This is number 4. What's number 4?
Medullary cavity.
Yeah. Yep. And this is number 2. What's number 2?
Diaphysis.
Exactly. Yes, absolutely. So in the diaphysis, you're going to find these structures. Let's use green. These tubes, because you're seeing a cut section of it, you don't see it as tubes, but imagine tubes running down. All over. What are these tubes called? What is this tube called?
Osteon.
This is an osteon. So now you should be able to, in your brain, comprehend how an osteon fits into a long bone. Okay. Is my video? I think my video is on, right? So if you imagine tons, so each one of these pens is a tube. Okay. So imagine tons of these tubes surrounding the diaphysis. So each one of these tubes is an osteon. Okay. So I pulled one osteon out right here. Okay. So now let's look at an osteon. An osteon actually looks identical to the diaphysis itself, but it is not the diaphysis. It's an osteon. It also has a center, but now the center is called what? Let's go with number 7. What's the center of an osteon called? It allows for blood vessels and nerves to move through it.
The central canal.
Yeah, good job. Okay. Then it also has these pockets. Let's do a different color. Let's do blue. Right, blue. So it has these little pockets. These little lakes. It looks like a little lake. And these little lakes contain osteocytes. What are these little lakes called? Let's call it number 8. What's number 8? What's number 8?
Lacunae?
Yeah, exactly. Now, the lacunae has these tubes that connect all the lacunae together. These channels, these networks. What are these channels or networks called?
Canaliculi?
Yeah, it sounds like an Italian dessert, to be honest. So we call it number 9, and number 9 is canaliculi. Okay, and if the term is ever confusing, what I do is that I just divide the term up, like canaliculi. And I do that a lot with the muscles to kind of help me remember how to spell it as well. Canaliculi. Okay. So you can divide it like that. So these are the channels that connect them. So the osteocytes that are trapped inside of the lacunae, they're cells, right? So they're going to metabolize, they're going to produce waste, they're going to need nutrients. So there needs to be a way to move the waste and nutrients back and forth, and it does it through the canaliculi. They're little channels that allow for things to move back and forth, but also for communication between the cells. Okay. All right. Then it's like a Twinkie, right? So the Twinkie kind of has these wrappings of the matrix. kind of like an onion. Think of layers of onion. Okay, so these layers of concentric matrix, what is that called? Like layers of an onion. What is that called? We don't have a lot left on our terminology list, right? It's not an osteon. We've already covered canaliculi, central canal. Go ahead.
Lamellae.
Yes, that's a lamellae. So the lamellae, I'm going to write it in a little bit darker color. The lamellae essentially is a concentric ring, rings of matrix. And what's the matrix made up of? Calcium, collagen. Okay. All right, so hopefully you can kind of see how these terms are connected to each other. I'm running out of time, so I'm going to speed through the rest so I can get to the two requested content. In the bones, there are 4 main cells you need to know: osteogenic, osteoblast, osteocyte, osteoclast. Know what they do, what their functions are, and how they're related to each other. So osteogenic cells become osteoblasts, and then osteoblast is the bone building cell. The B in blast is the B in building. So the osteoblast builds the bone matrix by pulling calcium, collagen, and phosphate all together until it mineralizes, okay? And then that traps the cell there, and it's still the same cell, but now it's not building matrix anymore. It's still the same cell, but because it has a different function now, we now call it the osteocyte. Okay, so osteogenic becomes osteoblast, and osteoblast becomes osteocyte, where the osteoclasts are a cell upon themselves. Osteoclasts are the bone reabsorbing, and that's because they have these structures right here that contain the ruffled border that contains lysozymes that break things apart. Okay, so ruffled border, lysozymes is associated with the bone reabsorbing cell, or otherwise osteoclast. Okay, you have 2 main bone types, tissue types. You have compact and you have spongy. So make sure you go through them. And then you also want to be able to know what's the difference between endochondrial ossification and intramembranous. For example, what's the starting material of endochondrial? Let's look at the term. Endo meaning it starts from the inside. Chondrial, whenever you hear the term chondrial, what should you think of? Like chondrocytes. What does chondrial mean? What tissue types contain chondrocytes? Cartilage. Okay, so that's why I always highly recommend doing a vocabulary sheet, because that helps you identify what things are. Okay, so whenever you see endo, inside, chondro, cartilage. Okay, so the starting material for endochondrial ossification is cartilage, where intramembranous, the starting material for this is mesenchyme. Okay, and that's why endochondrial ossification builds things like long bones, where intramembranous ossification builds things like flat bones. Okay. Here's an image from my in-class version, and I kind of go through the same thing. Okay, so kind of make sure you go through that. For joints doesn't have a lot of physiology, but it has a lot of anatomy and memorization, so a lot of terminology. It's really, really helpful, especially for joints, to do this cognitive map, because if you look at the terms, they all look the same. Like I struggled with this one because you had to know the difference between the terms for movement versus the terms for the actual joint examples themselves. So if the test question is, what kind of movement is this? You wouldn't answer sutures, gomphosis, or syndesmosis. You would answer synarthrosis. Okay? And if I say, what example of a fibrous joint that is found between the jaw and teeth, you would say gomphosis. That's when you would use these terms. Okay? And they sound the same: synarthrosis, syndesmosis, synchondrosis. You know, lots of similar terms, and that's why having this cognitive map helps you to kind of see the difference in the terms and whether is it a term for movement or is it a term for example, and if it's an example term, then what is an example of. Okay, so finish this up. I will always include this PowerPoint in your post. Questions? Another thing you want to know is make sure you know the synovial joints. Know these terms. What's a meniscus? What's a bursa? What's a synovial capsule, synovial cavity versus an articular cartilage? And I provided a really nice image of everything here. Okay. All right, so moving on to muscles. Muscles is a really difficult chapter. So you want to make sure that you set aside enough time to review this. Okay. It takes it's a process. This muscles is definitely a process. You're not going to get it in one day. It takes time, and that's why the more you're exposed to this content over a long period of time, the more it's going to make connections in your brain and make more sense. Okay, so if at first it sounds really like I don't understand this, it's not that you will never understand it, but it just takes time to keep kind of adding layers and information to it. Okay. So for muscles, make sure you know the function, the structure, what an NMJ is. What does NMJ stand for, by the way? What does NMJ stand for? Neuromuscular junction. Oops, oh no, my battery. Hang on. Junction. Okay, so the neuromuscular junction. And in that name, it tells us what parts there are, right? Neuro, meaning the neuron, muscular, meaning the muscle cell, and junction, because there's a space in between it. Okay? If you can see my screen, imagine this. Here's one. Let me get another pen.
Another pen.
So imagine this. So here's the neuron right here. Here's the muscle cell. Okay. And there's always going to be a space in between them. Okay. The brain is going to send a signal to the neuron in what form? How is signal transmitted from the brain to the neuron? It's a chemical, right? It's what we call neurotransmitters, right? So the brain's going to release chemicals that then bind to the dendrites on the neuron, and then that stimulates an electrical response, and that's where the action potential comes into play, right? Here's an electrical, and it just moves down, right? Action potential, depolarization, repolarization, depol, repol, depol, repol, depol, repol, depol, repol, and it continues on until it reaches the end of the neuron. What's the end of the neuron called? What's the end of the neuron called?
Synaptic knob.
Excellent. Excellent. So that's the synaptic knob. Okay. So the synaptic knob, so here's a wire, right? So because this wire has this plastic around it, I touch it, do I get shocked?
No.
No. But if the plastic wasn't around it, if my hand was right here, would I get shocked?
No.
No. What about right here?
No.
What about right here?
Yes.
What does that tell you? In order for electricity to transfer, it has to have what? Not here. Not
Contact.
here. Yes, you got it. For electricity to transfer, you have to have direct contact. But I just established that here's the neuron, here's the muscle cell. Do they ever connect with each other?
No.
No. And that's where you have the term neuromuscular junction. It's not just called neuromuscular, it's called neuromuscular junction because there's a space in between. So there's a space, which means the electricity can't transfer directly. And that's why at the synaptic knob, it transfers, it converts electrical back into chemical. And that's where you have the acetylcholine come into play. At the synaptic knob, the synaptic vesicles, when the electricity reaches the synaptic vesicles, it's going to cause the release of acetylcholine. The acetylcholine is going to cross over that junction or that space and then bind to the receptors on the muscle cell, and that's what regenerates the electrical impulse. Okay? So this whole process of the neuromuscular junction is because it's got to go from electrical to chemical back to electrical because they never come into direct contact. Okay? And that's the first part of the sliding filament theory. Right? Is the electrical stimulation. Okay? Then you have the contraction, and then you have the relaxation. Okay, so that's the sliding filament theory. Metabolism is a topic on its own. Okay, so because I did make a video on the neuromuscular junction and the sliding filament theory, you can find that video in the announcements page. As well as the metabolism, I'm gonna stop here for this so that I can move on to the nervous. Okay, so make sure you watch the video. Briefly though, this is to show you kind of the connection between muscle and the breakdown of muscle, how the muscle becomes the fascicles, becomes the muscle cell, the muscle fibers, and then you take a deeper look inside of the muscle cell or the muscle fiber. Okay, and that's where you'll find the sarcolemma, the sarcoplasmic reticulum, the myoglobin, the mitochondria, the glycogen, the myosin, the actin, and so on. Okay, so make sure you go through that. Okay, so here's the sliding filament theory. You can see it starts with excitation, and then excitation, contraction, and then contraction, and then after contraction, you have relaxation. Okay, and there's a video on this, so make sure you kind of take a look at that. Okay, moving on to the nervous system. For the nervous system, you want to make sure you know the division of the nervous system. So you can see that the nervous system is divided into the central nervous system, which includes the brain and the spinal cord. And then the peripheral nervous system essentially is all the nerves. Nerves are divided into two main types, sensory and motor. Under sensory, that means it's taking information from the environment and sending it to your central nervous system. They're divided into a subcategory of somatic and visceral. Okay, same thing for the motor. Motor is going to take the information from your brain and send it to muscles, glands, or another neuron, depending on how far it has to go. But these types of neurons are also divided into two main types, visceral and somatic. Somatic, we often think of as voluntary. It's like your skeletal muscle. You know, you have control over it. Where the visceral motor neurons are more of your autonomic nervous system, or otherwise known as involuntary. Now, the visceral or autonomic nervous system is divided even further to the sympathetic and the parasympathetic. The main analogy of that is your sympathetic is kind of your stimulated, right? You're either going to fight or you're going to flight or fight. So responses, like, your jerk, your reactions. Where your parasympathetic is more of you're resting and digesting. It's not that they can't happen at the same time, but usually one dominates over the other. Okay. And then in terms of your neurons themselves, you have tracks. Tracks are the bundles of nerves, and then the nerves are bundles of neurons. And the neurons consist of three main parts: the dendrites, the soma, and the axon. So make sure you know the parts of it. Then moving into your brain, make sure you know the regions of the cerebrum, as well as the other main regions of the brain, which is your cerebellum, your diencephalon, and your brainstem, and then the subregions of that. What else? Make sure you know the spinal cord. You have 31 pairs, 8 are cervical, 12 are thoracic, 5 are lumbar, 5 are sacral, and 1 for coccyx. I tend to remember, based on the cervical, the thoracic, the lumbar, and then the sacral and coccyx are kind of connected to each other. Okay. And make sure you understand the parts of the spinal cord. For example, what's the horns versus what's the columns, and how a neuron, whether it's a motor or a sensory neuron, moves through. Okay. All right, so here's one of the requests is, what is a nerve impulse conduction? So the neurophysiology. So the nerve impulse conduction, so we talked about briefly about how signals move along the neuron, right? How the brain sends it through chemicals, and then the chemicals bind to the dendrites, and then the dendrites carry it along the axon until it reaches the synaptic knob. Now we're going to dig deeper and be more detailed as to how exactly it moves along here. Okay? Questions? Sorry, I went through that really fast. Questions? Is it confusing? Is it starting to make sense? Would you like me to rephrase something, give you more examples? Silence makes me kind of nervous. 1, are you still there? Am I still online? 2, it means you're thinking about it real deeply. So that means you may have questions. Let's start with the first one. Is anyone still here?
Yes.
Okay. I was getting nervous. Questions? You can ask me to restate something too. I'll go back to a different slide and so that you can take a look at it a little bit more.
I think you're good to keep going.
Okay. Feedback is always appreciated. All right. Okay, so the nerve impulse conduction. We start off with resting membrane potential. Another way of saying resting membrane potential is polarized. When something is polarized, it means that it is negatively charged. Okay, so rather than just memorizing what I just said and not understanding what it means, let's break it down. Okay, so here's a neuron. Okay, so you know that the neuron on the inside, what ions would you have on the inside?
Potassium.
Say that with confidence. You had it right.
Potassium.
Yes, ma'am. Potassium. Okay. So that's positive. You're like, wait a minute. That's not making sense. If on the inside of a neuron I have potassium, which is positively charged, why is it negatively charged? Who can answer that? What else is inside of a neuron?
Proteins.
Yeah. There's large proteins on the inside. And these proteins do not move. Okay, so even though there is potassium on the inside, because of the proteins that are negatively charged overall, a neuron at rest, nothing's happening, reads negative 70 millivolts. And you know I don't usually give you numbers. So when I do give you numbers, that means you should know these numbers. Okay, it might be on the test. Okay. So it is negative 70 millivolts because you have the negatively charged proteins, even though you do have potassium on the inside. Then on the outside, what do you have?
Sodium.
Yeah, excellent. And then you have these gates or these channels called sodium-potassium pumps, which allow the ions to move back and forth. Okay, they're gates that open or close. Right? So at this point, there is no stimulation. Nothing's happening. And it's negative 70 millivolts. But let's say that the brain wants to send a signal. Okay, so it's going to release these chemicals, these neurotransmitters. And okay, the dendrites receive these chemicals, and it's going to trigger what's called a local membrane potential. A local membrane potential occurs along the dendrites and the soma. That's where the local membrane potential is. It only occurs along the dendrites and the soma. So the signal along here. Okay, so the signal causes a slight change, depending on how strong the signal is, a slight change in the membrane potential. Okay, so it's going to look something like this. So here's resting membrane potential. Here's the slight change in local membrane potential. Now, the thing about local membrane potential is that it is graded. In other words, you can have A, B, C, or D. So there's different strengths. It is reversible, meaning if it's not strong enough, it's not going to go. Okay, and as it moves along, it actually gets weaker. Okay, so the signal that's sent from the brain right here has to achieve a difference from this right here to here before it can reach the action potential that looks like this. So this right here is the local membrane potential. And in order for it to trigger the action potential, it must be at least 15 millivolt difference. Okay, which means from here to here, it's got to reach what? Negative what?
55.
Yeah, and that's a number you need to know as well. Okay, so either the 15 millivolt difference or it has to reach negative 55. If the local membrane potential is able to reach negative 55, that's when it achieves threshold. Okay, so the signal is strong enough to then move to here. Actually, that's not quite right. It just moves to right here. Okay. So it reaches threshold at the axon hillock. That's where this yellow line is, the axon hillock, when the signal is strong enough to be sent. Okay. Now at this point, all the channels along the axon is going to allow for a rush of sodium to move in. Okay. Now when sodium moves in, you had the potassium already. Now you have the added sodium. Even with the negative proteins, the value is going to go up to what?
Positive 35.
That's right. It's going to become positive, and it's going to go all the way up to 35 right here, 35 millivolts. At which point, it's going to flip the switch. Now, instead of potassium going in, now instead of sodium moving in, what's going to move out?
Potassium.
That's right. That's repolarization. And because potassium is moving out, it goes down again. Does that make sense? So now the question is, why does that have to happen? So the analogy I'm going to give you is this. Imagine you have a bowl of water, okay, and you put a plastic ball on top. It's floating, right? If the water is stationary, if it's stagnant, does that ball move?
No.
No. So what do you have to create in the water for the ball to move from one location to the next?
Waves.
That's what depolarization and repolarizing is creating. It's creating waves to push the signal along. Okay, so here's the neuron right here. Here's the signal. Depolarization, repolarization, depolarization, repolarization, depol, repro, depol, repro, depol, repro, along the whole length of the axon. That's an action potential. Until it reaches the synaptic knob, at which point calcium rushes in, causing the synaptic vesicles to push the acetylcholine out. Okay? Make sense? Did that answer your question about the neurophysiology? I couldn't remember who asked that. So you can see that here as well. You have from resting membrane potential. You can see that it's positive on the outside due to the sodium, negative on the inside. Even though it has potassium, it's got that negative, the proteins. So when an action potential happens, it has to go through threshold membrane potential first. But when an action potential is reached, you can see it goes from depolarization to repolarization, and that's what moves the signal along. Okay? So this is a really nice kind of wrap up of the nervous system and the electrophysiology. So you can see resting membrane potential here, local membrane potential here, how it reaches threshold, then action potential. And remember that action potential involves depolarization as well as repolarization. And I drew a map of it right here, of how you can see it goes from A to B to C to D to E. So what would you call A?
Resting.
Yeah, let me zoom in for you. Can you see it okay?
Yes.
Okay. So A is resting. What's B?
Local.
Yeah. So what's C?
Threshold.
Yes. What's D?
Action.
It's a part of action, but what specific part?
Depolarization.
Beautiful. That leaves E.
Repolarization.
Yes. Now, between depolarization and repolarization, it's going to end up being more negative than it was at the very beginning. And the reason for that is because sodium and potassium move at different rates. For every 2 sodium that moves in, 3 potassium moves out. So you're losing more positive than you gain. And that's why instead of going back to just -70, it actually goes below -70. We call that hyperpolarization. Polarization meaning negative, hyper meaning overly negative. Okay. So that's the hyperpolarization. That's letter F.